Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the Transport Across Membranes chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. But I'm going to be starting with the cell surface membrane. Okay, so the cell surface membrane surrounds the cell. It's mainly made up of what we call a phospholipid bilayer. This is two layers of phospholipid molecules facing away from each other. Each phospholipid has a phosphate head and lipid tails. The phosphate heads are hydrophilic, meaning they're attracted to water, and the lipid tails are hydrophobic, meaning they repel water. This makes the structure of the phospholipid bilayer make more sense. The reason both sets of tails are pointing inwards is because the tails are moving as far away as they can from the aqueous solution outside the membrane. The opposite goes for the hydrophobic heads, which point to the outside of the cell surface membrane as they're attracted to the water. Functions of the phospholipids in the membrane are to allow lipid-soluble substances to enter and leave the cell, to prevent water-soluble substances entering and leaving the cell, because, for those of you who don't know, water-soluble substances are repelled by lipids, and lipid-soluble substances are repelled by water. And the last function is to make the membrane flexible and self-sealing. Interspersed through the cell surface membrane are proteins. Some proteins are in the surface of the bilayer but don't extend completely across it. Other proteins completely span the phospholipid layer from one side to the other. Some are channel proteins and some are carrier proteins. Here are the functions of the proteins in the membrane: to provide structural support, to act as channels transporting water-soluble substances across the membrane, to allow active transport across the membrane through carrier proteins, to form cell surface receptors for identifying cells, to help cells adhere together, and to act as receptors. Another component that may be present in the cell surface membrane is cholesterol, which is embedded in the phospholipid bilayer. The functions of cholesterol in the membrane are to act as recognition sites, to help maintain the stability of the membrane, and to help cells attach to one another and so form tissues. Another component is glycolipids. They are carbohydrates covalently bonded with lipids. The function of the glycolipids in the membrane are to act as recognition sites, to help cells attach to one another and so form tissues, and to allow cells to recognize one another, for example lymphocytes to recognize an organism's own cells. The cell surface membrane controls movement of substances in and out of the cell. But many molecules can't freely diffuse across the cell surface membrane because they are not soluble in lipids, so cannot pass through the phospholipid bilayer. They are too large to pass through the channels in the membrane. They are of the same charge as the charge on the protein channels, so even if they're small enough to pass through, they get repelled. Or they may be polar or electrically charged, so have difficulty passing through the non-polar hydrophobic tails in the phospholipid bilayer. The cell surface membrane is often depicted using the fluid mosaic model. It's said to be fluid because individual phospholipid molecules can move relative to one another, which gives the membrane a flexible structure that is constantly changing shape. And it's like a mosaic because the proteins that are embedded in the phospholipid bilayer vary in shape, size, and pattern in the same way as stones or tiles of a mosaic. Now let's move on to the types of transport that can happen across membranes. One you're already familiar with is diffusion. Diffusion is the net movement of molecules or ions from a region where they are more highly concentrated to one where their concentration is lower until an equilibrium is established. The type of diffusion we know about is simple diffusion, which involves nothing but the substance being transported. Molecules that undergo simple diffusion are small, non-polar molecules, like oxygen and carbon dioxide. We can also have facilitated diffusion. This is still passive diffusion, but involves channel proteins or carrier proteins. Channel proteins form water-filled hydrophilic channels across the membrane, allowing specific water-soluble ions to pass through. The channels are selective, each opening in the presence of a specific ion. With carrier proteins, when a molecule like glucose, for example, that is specific to the protein is present, it binds with the protein, causing the protein to change shape in such a way that the molecule is released into the inside of the membrane. Osmosis is the passage of water from a region where it has a higher water potential, so a lower concentration of solute, to a region where it has a lower water potential, so a higher concentration of solute, through a selectively permeable membrane. Let's now look at the effect of osmosis on a plant cell. To do this, we look at the protoplast, which is the outer cell surface membrane, the nucleus, the cytoplasm, and the inner vacuole membrane combined. In hypotonic solution, the protoplast pushes against the cell wall, and the cell is said to be turgid. In isotonic solution, the protoplast no longer presses on the cell wall, so the cell is said to be at incipient plasmolysis. In hypertonic solution, the protoplast pulls away from the cell wall, so the cell is said to be plasmolyzed. Active transport is the movement of molecules or ions into or out of a cell from a region of lower concentration to a region of higher concentration using ATP and carrier proteins. Active transport differs from passive transport because the metabolic energy in the form of ATP is needed for active but not passive transport. In active transport, substances move against a concentration gradient from low to high concentration. Carrier proteins and molecules which act as pumps are involved, which is the case for facilitated diffusion but not simple diffusion. And the process is very selective, which is again the case for facilitated but not simple diffusion. The process of direct active transport of a single particle is as follows. The carrier protein spans the plasma membrane and binds to the molecules or ions to be transported on one side of it. On the inside of the cell, ATP binds to the protein, causing it to split into ADP and a phosphate molecule. As a result, the protein molecule changes shape and opens to the opposite side of the membrane. The molecule or ion is then released to the other side of the membrane. The phosphate molecule is released from the protein, which causes the protein to revert to its original shape, ready for the process to be repeated. The phosphate molecule then recombines with ADP to form ATP during respiration. Sometimes more than one molecule or ion may be moved in the same direction at the same time by active transport. Sometimes one molecule or ion is moved into the cell at the same time as a different one is being removed. For example, the sodium-potassium pump is where the ions are actively removed from the cell, while potassium ions actively are taken in from the surroundings. A good example of co-transport is the absorption of glucose into the ileum. Step 1, the sodium ions are actively transported out of the epithelial cells by the sodium-potassium pump into the blood. The sodium-potassium pump is a carrier protein which transports sodium ions one way and potassium ions in the opposite direction. This maintains a much higher concentration gradient of sodium ions in the lumen of the intestine than inside the epithelial cells. So, sodium ions can diffuse into the epithelial cells down this concentration gradient through a different type of carrier protein, or co-transport protein, in the cell surface membrane. As the sodium ions diffuse in through the second carrier protein, they carry either amino acid molecules or glucose molecules into the cell with them against their concentration gradient. So this is co-transport via a co-transport protein. It's the sodium ion concentration gradient that powers the movement of the glucose or amino acids rather than ATP, making it an indirect form of active transport. There's now a higher concentration of glucose in the cell than in the blood, so the glucose diffuses into the blood via facilitated diffusion. The last thing to cover are the required practicals. There are 2 required practicals in this topic, and we're going to start with required practical 3, which is the production of a dilution series of a solute to produce a calibration curve with which to identify the water potential of a plant tissue. Essentially, we put some potato in different glucose concentrations and see if they increase or decrease in mass. The first step of this practical is making a dilution series of glucose concentrations. We're starting with a 1 molar solution of glucose, and we want to create a 0.8 molar solution, a 0.6 molar solution, a 0.4, a 0.2 and a 0 molar solution. We want the total volume of all of these to be 20 centimeters cubed. So given this information, feel free to pause the video and calculate on your own the volume of glucose and water needed in each tube. Okay, so one way to do this is with the equation CSVS equals CWVW. So in the case of the 0.8 molar tube, CS is the stock concentration, which is 1. VS is the volume of stock needed, which is what we're trying to find out. CW is the concentration we're aiming for, which is 0.8. And VW is the end volume we want, which is 20 centimeters cubed. So VS will be 16 centimeters cubed and the rest will be water, so that's 4 centimeters cubed of water. If we do that for the rest of the tubes, these are the numbers we get. Next, use a cork borer to get 6 identically sized chips from the potato and block these with paper to remove excess water. Weigh each chip and record these as the initial masses. Place 1 chip into each solution we made in the dilution series and leave them for 20 minutes. Remove the chips from the solution, gently blot them with paper to remove excess water and reweigh the chips. There'll be a different mass now because they'll have either absorbed water or released water into the solution. If the potato is in a hypotonic solution, it will have increased in mass because the solution has a higher water potential than the potato, so water moves via osmosis into the potato. If the potato is in a hypertonic solution, it will have decreased in mass because the solution has a lower water potential than the potato, so water moves via osmosis out of the potato. If the solution is isotonic, the potato will have no change in mass. We can calculate the percentage change in mass of the chip by doing the final mass minus the initial mass all over the initial mass multiplied by 100. Then plot the percentage change in mass against the concentration to produce a calibration curve. This graph has a positive percentage change for increases in mass and negative percentage change for decreases in mass. At the point where the graph crosses the x-axis, the solution is isotonic. If we had a solution with an unknown concentration, we could do this experiment with it, calculate the percentage change in mass and using the graph find its concentration. Required Practical 4 is the investigation into the effect of a named variable on the permeability of cell surface membranes. Permeability is the ability of substances to move across the membrane. More permeable means a faster rate of transport and less permeable means a slower rate of transport. The 2 main things that affect permeability are temperature. In general, the higher the temperature, the more permeable the membrane. This is because higher temperature means more kinetic energy. The phospholipids have more kinetic energy, so move around more, making it easier for things to get through. However, at 0 degrees and below, the membrane is more permeable than you'd expect because the channel and carrier proteins deform, so more can get through them than usual. And the other thing that affects permeability is the solvent used. Because alcohol, for example, dissolves lipids, so the phospholipid bilayer would start to break down. So an increase in solvent concentration would increase the membrane permeability. So the independent variable we'll be using is temperature. What you're going to do is use a cork borer to make around 20 discs of beetroot. We're going to wash these discs, then label 6 test tubes with the temperatures we'll be using. So we've got 25 degrees, 35 degrees, 45, 55, 65 and 75. Add 10 centimeters cubed of distilled water to each test tube and then set up 6 water baths to the same temperatures stated. Put each test tube with a thermometer in it into the corresponding water bath and wait until the test tube reaches that temperature before removing it from the water bath. Place 3 beetroot discs into the test tube and wait for 60 seconds before removing them. a good idea would be to shake the test tube to evenly disperse the pigment. Then, using a colorimeter, measure the absorbance of the coloured liquid produced. If we plot a graph of absorbance versus temperature, it'd look a bit like this, because as we know, the membrane permeability increases with temperature, so more of the beetroot's pigment can diffuse into the water. And that brings us to the end of transport across membranes. Thank you for watching. Please like and subscribe. If you like this style of teaching and would like to get tutored by me or a member of our team, you can go to our tutoring website at thesciencebox.net. I'd also really recommend our Instagram and TikTok for tiny doses of revision. It makes you feel productive even if you're just scrolling TikTok, so that's a bonus.